I'm Ana Laura Hernández-Ledesma. I'm a PhD student at Universidad Nacional Autónoma de México, and I will present you our work entitled Allele-specific gene expression during tolerance induction on dendritic cells from Mexican women with systemic lupus erythematosus. Systemic lupus erythematosus is a chronic autoimmune disease with a multifactorial component that causes multisystemic damage and a wide variety of clinical manifestations among people with SLE. This disease mainly affects women in a ratio of 9 women per man. Higher prevalence and disease severity has been observed among African, Asian, and Hispanic descendants. Under health conditions, the immune system can distinguish between external antigens from pathogens and self-antigens through a set of processes called immune tolerance. Although the physiopathology of SLE has not been fully established, it has been proposed that the convergence of different factors, such as the increase of apoptotic cells, the exposure of cellular components, and the formation of immunocomplexes, favors the identification and presentation of self-antigens and the subsequent production of autoantibodies. This continuous stimulation of the immune system can trigger an exacerbated immune response, which in turn leads to the loss of immune tolerance. Tolerogenic dendritic cells, TOL disease, are a group of immune cells characterized by its anti-inflammatory phenotype. These cells can polarize the T-cell differentiation to Tregs, which can inhibit active immune responses. This ability to modulate the immune response has made tolerogenic cells a target of interest for the development of potential immunotherapies. Genetic factors are important on the SLE development. Heritability has been estimated between 40 and 60%. Although more than 90 loci have been associated with SLE, there is still scarce information underlying the involved genes and mechanisms in this disease. Allele-specific expression is a gene regulation mechanism characterized by the overexpression of one allele over the other, which can be associated with physiopathological process. The aim of this study is to characterize the allele-specific expression during tolerance induction of dendritic cells from Mexican women with SLE. Participants were identified from the Mexican Lupus Registry, an online platform with self-applied service designed to advance into the knowledge and understanding of the characteristics of the Mexican people with SLE. 21 SLE volunteers and 9 healthy controls were included in this study. Informed consent was provided and all the data were anonymized. 2 samples were obtained from each participant, blood and mouth exudate. From blood sample, peripheral blood mononuclear cells were isolated using lymphoprep density gradient. Afterwards, monocytes were isolated using EasySep human monocyte isolation kit from Allele. Monocytes were then seeded into 2 wells. One was stimulated using GM-CSF and IL-4 to differentiate them into monocyte-derived dendritic cells or modices, and the other one was stimulated using GM-CSF, IL-4, and IL-10 to differentiate them into tolerogenic dendritic cells or TOL disease. Cells were incubated and stimulated for 7 days. Then, in order to activate dendritic cells, modices and TOL disease were separated into 2 wells. One continued with the corresponding stimulus, and in the other well, additionally to the corresponding stimulus, imiquimod was administered. Cells were recovered 24 hours afterwards for RNA-seq and flow cytometry. Changes in surface markers associated with monocytes, dendritic cells, and antigen presentation were evaluated using flow cytometry. RNA was recovered from cell culture using RNA-seq microkit from Allele. Library preparation and RNA sequencing will be performed by Novogene. Quality control analysis, alignment to reference genome, transcript assignment, and differential expression analysis will be performed. Genotype will be determined using the Infinium Global Diversity Array 8, making the corresponding pre-imputation quality control analysis and genotype imputation. Allele-specific expression will be determined by performing the corresponding alignment filtration, allele count, and identification of sites with allele-specific expression. 21 volunteers with SLE and 9 controls have been sampled. General characteristics are presented on Table 1. In Figure 1, we can observe the percentage of cells expressing CD14, a marker used to identify monocytes, and the percentage of cells expressing CD11C, a marker used to identify dendritic cells populations, which increases significantly through the cell culture after stimulation. In Figure 2, we present the co-expression of surface markers involved on antigen presentation. No difference were observed between SLE participants and healthy controls on expression of these markers. DNA has been isolated and samples for RNA-seq have been obtained. CD11C expression changes after cytokine stimulation. This could be indicating an adequate differentiation process to dendritic cells. In line with previous studies, no significant changes were observed between SLE and healthy controls on the expression of markers associated with antigen presentation. There is needed to define the factors that could be contributing to dendritic cells' tolerogenic capability on SLE. RNA-seq and genotyping analysis will allow us to identify allele-specific expression patterns associated with the tolerogenic potential of dendritic cells from Mexican women with SLE. Thank you for visiting my poster. I will take any further questions.